Next time, Ron, can we switch the order? Okay, so we're going to talk about the perception of voice. And so far, let's see here. Figure out. Great. So far, we've learned quite a bit about anatomy and physiology. We've learned about production in a variety of different ways. A great acoustics lecture with audio demos, man-made audio demos, which were awesome. And now we're going to talk about how the auditory system makes physical and physiological transformations of those acoustic signals, and the brain forms the perception of voice. So we're going from the vocal production mechanisms to the acoustics to the auditory system, and that's why the auditory scientist gets to follow STEM. So let's think about perception in a very general sense. A simple definition is the process of attaining awareness or understanding of sensory information. That is a very generic definition, and actually when you think about sensory information, it includes vision, taste, smell, touch, hearing, perhaps a sixth sense. I think STEM has one. Balance could be a sixth sense as well. And so it's a very, very broad field. Some of us spend most of our careers focusing on a very narrow part of the field of perception, in my case included. But in terms of the perception of voice, the main sensory component is hearing. We hear spoken voice. And so we ought to think a little bit about the hearing mechanism. We won't go into too much detail. Some of you probably teach about this in some of your classes. I'm sure many of you had this in courses that you've taken. So I'm just going to give you a cursory overview of the auditory system, starting with the most peripheral part, the part that's on the outer part of the head. So normally we divide the human ear into three divisions: the outer ear, the middle ear, and the inner ear. And the outer ear starts with the pinna, and most people say the pinna funnels sound into the ear canal. Well, actually it does a whole lot more than that. The curvature that we see in the pinna ear functions as a delay and add filtering network that produces a comb-filtered sound. And I'm pretty sure we could have seen a comb-filtered sound on the spectrogram that was up here for the last presentation. And the shape of the spectrum changes depending on whether the sound is coming from up here, down here, or even lower. So the pinna actually tells us something about sound elevation by the way in which it filters sound. You just thought it was there to hang glasses, hearing aids, and jewelry. But it's actually serving a very important function for auditory perception. Then there's the ear canal, and the ear canal is a tube. We know everything there is to know about tubes already. What is the number one thing a tube produces? A resonance. And in the adult human ear canal, it's around 3,000 hertz. And for those of you who are speech scientists and know about the intelligibility of speech, many of our consonant sounds have key spectral features in that 3,000 hertz range. And so it's actually amplifying sound in an important speech range. So that's the outer ear. Then we have the middle ear. And the middle ear consists of the eardrum and three little bones in the middle ear, the smallest bones in the human body. And so we'll start the quiz. Normally I teach Monday morning at 8 o'clock and have a quiz every Monday. This is late, but the same phenomenon is going on. Everybody's tired. So let's have a quiz. What are the names of the three smallest bones in the body there in the middle ear? Malleus, incus, and stapes. Very good, very good. I'd say about 40% of you passed the first question. What's the nickname of those three? Hammer, anvil, and stirrup. And the stirrup is the easiest one to see there because it really does look like a stirrup. The hammer, kind of like a club. And the anvil, most people don't know what an anvil is anyway. So that's the middle ear. And then there's the inner ear, which we think of as the cochlea or cochlea. But actually it includes the semicircular canals that make up our balance system as well. But we're going to ignore that today. So if we focus a little bit more on the next slide, the outer and middle ear, what we're seeing now is condensation and rarefaction of air molecules produced by a sinusoidal source. You've heard all of those terms in today's talks. And so those are the red fluctuations in air molecules that then hit the eardrum and cause the eardrum to vibrate. So we've just transformed the sound from a vibration in air to a mechanical vibration of the eardrum, and the ossicles, the three bones behind the eardrum. And you can also see some muscles and tendons hanging out there. You can clearly see the stirrup, and you can see that it's pushing on a white open space, which would be the inner ear or the cochlea. It turns out that this transduction in the middle ear functions effectively as an amplifier, about a 30 to 40 decibel amplifier, which is pretty amazing. Most people thought that was just a place for infections. It's not just a place for infections, it's actually a mechanical amplifier by the area ratio between the eardrum and the foot plate of the stapes and the lever action of the three ossicles. So it's a mechanical amplifier. Why do we need to amplify sound there in the middle ear? Because the sound in air is vibrating with a certain amount of energy or amplitude or intensity, and it needs to vibrate the fluid that is in these little chambers in the cochlea. So we need to get a vibration from air to fluid. Think about sound above a pool going into sound when you're underneath the water in the pool. Most of the sound vibration hits the surface and bounces back off. Not much goes into the pool. And so you need an amplifier to get vibration into the pool of water that's in these two chambers of the cochlea. Now the cochlea, this is a human cochlea next to a penny, so it's fairly small. You can see that it's sort of a snail-shaped organ. The name cochlea comes from the same root word for the conch shell. And most of the time when we try to understand what's going on here, we take a slice. And so this is just a slice or a cross-section of the cochlea. And it has three chambers. The upper and lower chamber, both green, have the same kind of fluid in them. It's kind of like salt water. It's called perilymph. In the middle chamber, it's affectionately known as the scala media, has another type of fluid that is like cerebrospinal fluid. So it's a chemical compound that is very good for transducing electrical signals, which is actually what's going to happen in this little part of the cochlea. So another way to start to think about the cochlea is to sort of unravel that 2 3/4 turns of the cochlea. And we normally do that and then take a look at patterns of vibrations that can be set up on the cochlea. And so back over in the little triangle region here, this portion of the scala media is called the basilar membrane. It's the base, the basilar membrane. And sitting on top of it is really the sensory organ within the inner ear. But we usually just collectively call that the basilar membrane. And so if you uncoil that entire cochlea and stretch it out, it looks something like the little animations down below. And the pink regions span the width of the basilar membrane. Notice that here, here it is a fair, my hands are amplifying my pre-Parkinson's, I guess. You can see there, it's a fairly narrow region of the basilar membrane. So this organ actually functions like a stringed instrument, very much like a harp, or if you think of a grand piano, where towards the base, the fibers that make up the basilar membrane are short and tight. And if you think about strings that are tuned so that they're tight and they're relatively short, what is the pitch that they will produce? High pitch. Exactly. And so that portion, the basal portion of the cochlea, produces or transduces high pitch vibration into a mechanical and then an electrochemical sound that goes up to the brain. Likewise, if you see on the lower figure there, if you have a low pitch sound, it produces a vibration along the part of the basilar membrane that has longer fibers stretched across that have much more mass. So we have a stringed, tuned instrument within the cochlea, and it obeys all of the same vibratory principles that we learned about in the previous talk. In addition to that, if we start to look at the little area of the basilar membrane here that's in the rectangle, we can highlight some cells. There are 4 little purple cells there, and they have little cilia sticking out of the top, and those are known as hair cells. The cell bodies are the purple part, and the hairs are the little cilia on the top. The one to the far right that's not in the box is actually the hair cell that sends information up to the brain. And the 3 that I've highlighted in the box actually get signals from the brain to tell those cells to either get taller and skinnier or shorter and fatter. And so we have an active amplifier. The cell movement of those outer hair cells actually serves as another amplifier and a tuning device within the basilar membrane within the inner ear. So it's a pretty complicated system, and of course this is just a cursory overview, but what you can see is it obeys all the same principles of acoustics that we've learned and that apply to production, to transduction, and to now hearing. Finally, the sounds come out of the cochlea through these little orange fibers here. Those form a bundle called the auditory nerve, and the auditory nerve has a number of properties that are very important as well. As the sound level gets higher, the auditory nerve fibers fire faster or more frequently, more firing spikes per second. So we have a code of loudness in the firing rate of the auditory nerve fibers. But perhaps more important is the timing of that firing. It has very, very precise timing. And that's what allows us to encode some of that frequency information that we were visualizing and hearing in the previous talk. So timing is very important. In fact, the auditory system is by far and away the most temporally precise sensory system in the human body. Then we go from the auditory nerve to the central auditory pathway, and I'm not going to spend a lot of time on that, but believe me, it can get very complicated very quick, and we're still actually learning about how the central auditory system processes the input to form perception. But basically, the brainstem is super important in coding timing information, frequency information, and level information. The midbrain retains that same code and starts to integrate information, including integrating sound from the 2 ears. The thalamus starts to get information from other parts of the brain and actually is where audio-visual integration starts. And then our perception is formed in the cortex. So there's a lot going on in order to perceive voice. This is just a neat little demo. This is some electrodes put on this cartoon's head. And the electrodes are measuring brain electrical activity while this person is listening to the stimulus, which is the vowel ah here. And what you can see from the electrical recording from the brain is also a very periodic sound that has a lot of the same characteristics of the external stimulus that was played through headphones. In fact, the temporal precision of the neural code is so good that if we play the audio of that recording of brain activity, you can hear the vowel. And you can actually hear musical instruments and you can hear melodies by playing the evoked potential from brain activity. So the temporal precision is amazing. Okay, so let's get back to perception. We know a little bit about what the auditory system is doing. There are a few fundamental abilities that give us auditory perception. We have to be able to code certain parts of the sound stimulus in order to perceive things like voice quality or pitch or loudness. And fundamental abilities include detection threshold, the ability to detect that a sound has occurred, the ability to encode things like intensity or temporal characteristics. And in fact, our precision is amazing. If we have a sound of a certain level, and you want to know how much higher in level you have to make it in order to detect a change, we can detect less than 1 dB change in sound level. We have 120 dB dynamic range, so that means we can detect maybe not exactly 120 discrete steps, but many, many, many discrete steps in level from detection to uncomfortably loud. Our temporal precision we can measure in many ways, but it's amazing. If you look, milliseconds were mentioned earlier. Those are 1,000ths of a second. The left ear and the right ear can compare the time of arrival of sound on the level of millionths of a second, microseconds. 1 millionth of a second timing differences. Extremely precise temporal code within the auditory system that gets transduced up into the brain. And the brain can say, if a sound comes 15 milliseconds late to this side relative to this side, the sound must have come from over here because it got here first and got there later. It's pretty amazing. Spectral processing allows us to code the shapes of those formants that we saw in previous spectrograms, and spatial processing allows you to know that I'm coming from here, and my voice is coming from up here, and not over there, and not over there. Actually, probably not over here, because we have loudspeakers planted in the room-like spots. Okay, so in addition to these fundamental abilities, they lead to perceptual attributes that we actually think of much more often than fundamental auditory ability. And those perceptual attributes include loudness, pitch, and aspects of voice quality or sound quality in terms of general sound quality, tonality, roughness, and sharpness. So I'm going to spend just a couple of minutes on perceptual attributes highlighted here. Loudness. Loudness is something you kind of have an intuitive feel for, but you probably wouldn't be surprised if I told you there's a lot more to loudness than meets the ear. It's actually quite complicated. The definition is a simple definition, that auditory sensation in terms of which sounds can be ordered on a scale extending from quiet to loud. A very boring definition. And we have lots of ways to measure the perception of loudness. We can measure using rating scales. We can rate the loudness of different sounds. We can estimate the loudness of different sounds. We can match the loudness of one sound to another. In fact, back in the 30s, they developed the standard scale for loudness that we still use today, and in a few minutes I'm going to argue we might be able to leverage to help us better understand voice quality. So this is an interesting, very, very complicated figure that describes our loudness perception in one way. The x-axis is sound frequency from low to high. The y-axis is sound level from from low level to loud level or high level. And these contours are called equal loudness contours. Equal loudness contours. So every point on any one of those given lines has exactly the same loudness, even though the SPL value on the y-axis varies a lot. So for low-level sounds, say 10 decibels above the level that you can just barely detect, a sound that is only 10 decibels loud, 10 decibel sound pressure level, is equally loud as a 100 hertz sound that's 46 decibel sound pressure level. So they have the same loudness but very different SPL. So what that is telling us off the bat is you can't equate sound pressure level to loudness. Sure, the higher the sound pressure level, it's likely to be louder than a lower sound pressure level. But in this case, 46 dB sound pressure level of 100 hertz is the same as 10 dB sound pressure level at 1,000 hertz. So sound pressure level is not equal to loudness. Also at very high levels, like 80 dB SPL at 1,000 hertz, a sound at 100 hertz is equally loud when it's only 82 dB SPL. So those curves get flatter as we get higher in level. What does that mean practically? Let's think about listening to music. If music is recorded at a relatively high level, and I would argue most music is recorded at a relatively high level, we have very, very flat loudness contours. And we get what we would consider a full, rich reproduction of sound if you play it back at a high level. If you play it back at a low level, then you get the loudness of mid-frequencies much better than you get the loudness of low frequencies, and the sound does not seem very full or very rich. That's because of our unequal loudness contours at lower levels. So you might remember old stereo equipment used to have a little button called a loudness button. And you pressed that when you were listening at softer levels so that it would bump up the bass and give you a richer sound. You don't want to leave that pressed in if you go to high levels, where it's essentially like cranking up the bass. All because of these equal loudness contours. Okay, so back in 1936, S.S. Stevens said, how can we compare the loudness of one sound to the loudness of another sound? We need some standard loudness units. And he created a standard loudness unit called the SON, and it created the SON scale. One SON is equal to the loudness of a 1,000 hertz tone at 40 dB SPM. And now what you can do with this standard unit is you could compare the loudness of that 1,000 hertz tone to the loudness of my voice, to the loudness of this little hum I hear in the room, to the loudness of a refrigerator, to the loudness of a projector fan, and we have the ability to characterize loudness independent of the type of sound by referencing it to the standard unit of 1,000 hertz, 40 dB SPM. I make this point not because you care that much about loudness, but because I'm going to come back to this with respect to learning more about sound, voice quality. Is it used? Is the SON scale used? Well, I went to Home Depot's website, and I thought, what is an annoying sound? A bathroom fan. So I looked at 3 different bathroom fans, and I clicked compare and compare and compare, and then I did the comparison, and I put them in a little table like this for me, and I did a screen grab. And I've got 3 bathroom fans here. Notice the price, $31, $139, $240. Which one are you going to buy? Bathroom fan, $39, right? You don't care that much. The bathroom fan that's $31 has a sound level of 2.5 SONs. Its loudness is 2.5 times a 1,000 hertz tone at 40 dB SPM. 2.5 times as loud. You pay another $100 and you can get that sound level down to 1 SON, about 40 dB SPM. If you want to double that, you can get it down a little bit more, but there's a point of diminishing return. So we actually do use this loudness scale. And you probably didn't know that, but next time go to Home Depot, entertain me, go over to the bathroom fan section, look on the box, and it will have loudness in SONs. So will your dishwasher. So if you get a cheap dishwasher, it's going to be loud, have a lot of SONs loudness. If you get a nice Bosch, brand new, for the first year it's super quiet, year 2, a little louder, year 3. Okay, so how could we use such a scale clinically? Right now, we'll talk a little bit more about ways of measuring voice quality clinically in a minute, but we often use rating scales, which are very, very handy, easy to do, quick, give us useful information, but it's really hard to say if someone's breathiness was at 0.5 before surgery and at 0.3 after surgery, how much improvement you got from a rating scale. You can't really quantify it. But if we had a standard scale like this, we could say maybe pre-surgery we have 2.5 perceptual breathiness units. And 4 months after surgery, maybe only 1 perceptual breathiness unit. If we had this standard scale, we could actually do that. And then say, we've improved breathiness by a factor of 2.5. Insurance companies might be willing to give you some money if you could show that. Okay, that was loudness. Now we're on to pitch. With pitch, most people have a very, very good understanding of pitch. How many of you are musicians? Okay, so I shouldn't be talking to you about pitch. I'm a dumb hearing scientist, right? Well, there are 3 components of pitch, and most of us recognize the first 2. Many people haven't thought much about the 3rd one, but pitch height is what we most often think of when we think of pitch. That attribute of auditory sensation in which sounds can be ordered from low to high pitch. And on this little diagram here, we have low pitch going up to high pitch, and we call that pitch height, as opposed to pitch chroma, which reflects things like the notes of a musical scale, perceived as repeating every octave as you go up. For example, there are multiple C's on a musical scale. There's middle C, there's a C below middle C, there's a C above middle C. They repeat every octave, and that's pitch chroma. And then there's pitch strength. And pitch strength is the scale on which pitch can be estimated from faint to strong. And most people don't think about that. But you can have a very strong pitch percept or a very, very weak pitch percept. And that's reflecting differences in pitch strength. So let's see. I'm not sure if I'm going to be able to play this third sample here. Would you be able to just... You heard 4 sounds. The first 2 changed in pitch height. The second 2 changed in pitch height exactly the same amount. But the first 2 had a strong pitch strength, and the second 2 had a very weak pitch strength. Could you play that one more time? Can you hear the difference in pitch strength? So we'll talk about pitch strength in a minute. Come back to that now that you are well educated about the 3 components of pitch. So in sound in general, in auditory perception, there are 3 basic sound qualities. There are many, many qualities, but sort of 3 primary sound qualities: tonality, roughness, and sharpness. And what I'm going to argue is there are also many aspects of voice quality, but there are 3 primary voice qualities that we think about as speech-language pathologists, many of you professional musicians and singers, those of us who pay attention are none of the above: breathiness, roughness, and strain. And I'm going to argue that breathiness, roughness, and strain map onto the basic sound qualities of tonality, roughness, and sharpness. So let's think about perception. Let's get into that as we wrap up today. There are many different types of perceptual tasks and many ways to measure the perception of sound. You can classify different sounds. You can rate them on a scale from low to high, or 1 to 8, or on a 100-point visual analog scale. You can estimate their magnitude. You can match one sound to another sound. There are lots of different ways to measure sound, and that's called psychophysics. And we might want to know something about breathiness, roughness, and strain. And the most common and currently most useful, most widespread technique, certainly in the clinic, but also in laboratory studies, of voice quality uses a rating scale. The most common measure today is from the KV, where we can rate a number of different quality attributes as well as pitch, loudness, and other attributes. But one of the challenges with rating is that agreement among different raters can vary a lot, making it difficult to compare one rater's perception of voice quality to another rater's perception of voice quality. It can also vary over time, and it can be influenced by a number of things listed here, not the least of which are just rater biases. I might rate the size of those 3 boxes as 2, 3, 4. You might give it a rating of 4, 5, 6. Who's right or who's wrong, we don't know, but when we average them, we get something different, and I've got a mess right. Also, it may depend on set size. If we have to rate the size of the orange boxes, say I have to rate the size of the 4th orange box, I might give it a 2. But if I have to rate the size of the 4th green box, I might give it a 1. But they're the same size. So I've been biased by the set that they're mixed with. But it's the same size box. It might be the same voice, but I might be biased by previous voices that I've heard or future voices that I've heard, or voices that I've heard. So there's some objective measures, lots of objective measures that one might use to quantify voice characteristics or voice quality. The problem is that they often have a poor correlation with our perceptual data. So they aren't predictive of perception. They may be very informative about the vocal signal, but they're not telling us as much as we would like about our actual perception of the voice. And so we've taken a slightly different approach. We call it a psychoacoustic approach, where we borrow principles of auditory perception from the hearing science world, laboratory methods that have been used to study hearing, and computational analyses that take into account all of those transformations I talked about as the sound goes through the ear canal, the middle ear, the cochlea, the auditory nerve, and up through the auditory system to form a person. And so the approach is quite different from a simple rating of quality or just computing something from software. It's actually thinking about the perception of the sound using more rigorous psychophysical methods and borrowing, if not stealing directly, from the hearing science work. And so we also often use a single variable matching task, which allows us to play an acoustic signal, the voice that we're interested in, some comparison, and we vary some single variable of that comparison sound until it matches the voice of interest. And now we have a comparison that is known, has acoustic properties, and we can use it and its units to try to quantify that standard voice. That's called a single variable matching task. And then we take that acoustic signal, we put it through mathematical computations that reflect all of those processes going on in the auditory system to try to predict the perception. And I'll just show you a little data from that sort of thing. This is for breathiness. And what you see here are 11 stimuli on the x-axis going from very, very breathy to not too breathy. And on the y-axis, perceived breathiness in this single variable matching task where the variable was a sawtooth wave, which we heard about, mixed with noise. And what we see is we were able to measure with very high precision voices that ranged from extremely breathy, the signal to noise ratio, the ratio of the harmonics to the turbulent noise, background noise, was almost 1-to-1, giving you 0 dB, to very, very clean voice, unbreathy, least breathy, where the signal to noise ratio is 25 dB. And so we're using this matching task to characterize the breathiness. But notice we're not just rating them, we actually have units, decibels, signal to noise ratio, which allows us a quantity that we can then describe, we can understand, and we can try to predict through modeling. Just go to the pitch strength model, which brings us back to pitch. The pitch strength model is the notion we could model the breathiness in normal and pathological voices for people who are synthesizing breathiness by artificially creating a breathy voice like Lon was doing earlier. We can model that with a series of computations that reflect what the auditory processing does and a comparison to a noisy sawtooth wave. And here we have the original matching data I showed you on the previous slide. Here are the predictions of the perceived breathiness of these 11 voices with an estimate of pitch strength from a mathematical model. And you can see we can predict 87% of the variance in the perceptual data by doing that. I would argue that it's very difficult to make that good of a prediction with any simple transformation of the acoustic signal. So I know I'm red here. Let me move forward on the same thing works in running speech. And so to summarize, perception results from a very complex interaction of multiple acoustic cues and multiple auditory processes. It's not simple, it's pretty complex. The major attributes of the sound are pitch, loudness, and quality, where quality is breathiness, roughness, and strain for voice, but tonality, roughness, and sharpness for sounds in general. There are a number of objective measures, but we suspect that there are perceptually or biologically inspired models that will help us predict aspects of voice in a better way than many of our simple objective pictures. And with that, I'd better let our next speaker